So welcome to返學 DSE. So this is the DSE biology program and it's chapter 3 intensive video. In chapter 3, we will talk about cell membrane. And for the cell membrane, we have a model name which called the fluid mosaic model. And for the term fluid, it refers to the phospholipid bilayer, which allows the phospholipid molecules to move laterally, that is from left and right. And also for the term mosaic, it means that the proteins will interspan along the phospholipid molecules. And here is the general structure of the phospholipid bilayer. It will have a phosphate group, which means that it is a hydrophilic acid like water. And for it will have two fatty acid tail, and it is a hydrophobic region, which means it hates water. So that's why, because both outside and inside of the cell, there are majority will be aqueous medium, which means water. So the hydrophobic tails will point inward and form a layer of hydrophobic region within the bilayer structures. And as previously discussed, that there will be proteins in the cell membrane. So here are the 5 types of proteins. They are called the channel proteins, carrier proteins, receptors, antigens, and also enzymes. And this is the beginning of the study of the receptors and also antigens. So please kindly remember the general terms. So for the receptor is to turn on certain cell functions. And for the term antigens, it is required in the cell recognition in the body defense. And we will focus on the channel proteins and carrier proteins in this chapter first. And for the enzyme, it will be discussed later on in chapter 5 and 6. And how these structures relate to its functions. Because the core part of the bilayer is hydrophobic, so the small nonpolar molecules can dissolve in the bilayer and move across the membrane. And for small polar molecules, including ions and also glucose, they have to use either channel proteins or carrier proteins. And for large molecules like starch, it has no passage across the cell membrane. And there will be generally 2 factors affecting the permeability of the cell membrane. One is the temperature. As the temperature increases, the molecules move faster and it leads to a higher permeability. However, if it is boiled, it will lead to the damage of the cell membrane, and then it will lose the selectively permeability. It will become fully permeable, similar to the organic solvent, because it can dissolve the membrane. So at the end of the day, the membrane will become fully permeable. And there will be 4 types of movement across the membrane. You should use these terms whenever you are answering questions, and you have to discuss with others professionally. That is diffusion, osmosis, active transport, and phagocytosis. For me, the first 3 terms are more important than the last one. And for the diffusion, it happens for all substances except water. And it is a passive net movement of particles down the concentration gradient, which means that things will from a higher region to a lower region, which is like a slope, okay? So with a deeper slope, larger surface area, shorter distance, high temperature, smaller particles, and non-polar nature, this will all lead to faster diffusion rate. So what is the importance of diffusion? Because it allows exchange of materials within the environment, and it is involved in many processes, like the absorption of nutrients in the human small intestine. Because in the lumen, that is with the food, the nutrient content is very rich, so it is a very high in the concentration gradient. And comparing with the cell, there is a concentration gradient, so there is a net movement of nutrient from the lumen to the cell. For osmosis, it is a specific type of diffusion which is only for water molecules. And to understand it more, we have to understand the term water potential. With an increased solute concentration, there is a decrease in the water potential. And for the highest possible amount of water potential will be 0, which is pure water. So you may notice in this number 9, there is no number beyond 0. There's nothing on the right hand side, but everything is on the left hand side. So with an increase of solute concentration, the water potential will be more negative, which is in the here. And here is a general discussion about the osmosis. And because there is a selectively permeable membrane in between separating 1 and 2, the larger molecules, that's the purple circle, cannot pass through, while the water molecules indicated in pink can pass through it freely. So you may notice that pink can move, purple cannot move. And because the water movement is a random process, you may notice that there are 3 arrows from left to right and 1 arrow from right to left. So there is a net movement from left to right. And if you count the purple circle in 1 and 3, there will be no difference because 1 is actually 3. So you may notice that there is no change in the total number of the solute. There's the purple one which cannot pass through the cell membrane. And because of the net movement of water from the left to the right, you may notice that the concentration between 3 and 4 is eliminated when comparing with 1 to 2. So the general definition for osmosis is a passive net movement of water across a selectively permeable membrane based on water potential. It is from a region of less negative to a more negative region. So when comparing 10% sucrose and 1% sucrose with a selectively permeable membrane, there is a net movement of water from the 1% side to the 10% side. And you may also wonder, what if there is no membrane in there? So you may bring this question back to your class and discuss with your classmates or teacher. And here is the classic experiments. Usually we will either use the ionizing tubings or the potatoes to understand how osmosis works. Here is a ionizing tubing. You may take it as an artificial version of the cell membrane. You may notice that the protein cannot move across the membrane. So here is the beginning and here is the equilibrium one. The protein always inside. And there is a diffusion of the labeled reagent blue from inside to the outside, while for the red one is moving from the outside to the inside. Because both of them, that is the blue circle and also the red circle, can both affect the water potential. So there is probably no water potential difference. That's why the water level is not increased. So what does it mean to animals like us and to plants for osmosis? For animals, in a hypotonic solution, that is the distilled water version, the water will enter by osmosis, and that's why our cell will swollen and eventually burst, so the cell may die. And for the animal cell in a hypertonic solution, let's say in 10% sucrose solution, the water will leave the cell by osmosis, and the cell will become shrunken or wrinkled. Similar case will not happen in the plant cell because plant cell will have cell wall. So in a hypotonic solution, water still enter. However, the cytoplasm will push against the cell wall, which is rigid, and it will lead to a turgid cell. The term turgid you should bear in mind. And for the hypertonic solution, the water will leave the cell by osmosis, and the cell will become viscous and plasmolized because the cell membrane and the cell wall are separated from each other. And you may also notice that the vesicle will shrink. So what does it mean to the plant? When water is plenty, and then the cell will become turgid and it gives support to the cell and also leaf. That's why the leaves will stretch out. And when the water supply is limited and the cell cannot give the support, and that's why the leaves will drops. Okay. And plasmolysis is usually reversible. That's why the plants can survive for a while without enough water. That's why in the midday, the plants may look a little bit weak because of this condition is happening. And for the active transport, it will always require carrier proteins, and it is possible to be down the gradient. However, the main usage will be against the concentration gradient, which is like riding a bicycle. So when you're going down in the slope, you may either apply force or not apply force. However, if you are going upward, you have always to input some energy. And what is the source of energy? Will be the mitochondria. And its function is to take up additional useful substance from the environment. How about phagocytosis? Phagocytosis is usually taking up large particles directly, and it is also an active transport requiring energy, which is the same as the previous one. And for the changing of the shape, through the changing of the shape of the cell membrane, they will fuse together, and then it is phagocytosed. So what is the importance of phagocytosis in our body defense of red blood cells, white blood cell? The white blood cell will engulf the pathogens and eliminate them. And for unicellular organisms, they use this method to take in food. So here is an extract of the HKEA document. You have to understand what is diffusion, osmosis, and also active transport. Probably also where the phagocytosis is happened, and you should be able to explain plasmolysis and also hemolysis. Hemolysis is a term to describe when your red blood cell is damaged or destroyed based on the hypotonic solution. Then without any red blood cell, you may not and you cannot carry the oxygen and will lead to death. And it is also known as water poisoning, 水中毒. Okay, so here is the end of chapter 3. So see you next time.